Welcome back, folks. Today we have glycolysis part 2. So last time we were together, we learned about glycolysis. We learned some basics about each of the reactions. Well, today we'll start to talk about regulation of glycolysis, and this is definitely going to be our point of focus. We have some learning outcomes today. We're going to learn about anaerobic fermentation. So we're going to understand when a cell would choose that type of metabolism rather than oxidative metabolism. We're going to be able to describe the general mechanism of lactate dehydrogenase and describe how its R groups help. We'll understand why neither hexokinase nor pyruvate kinase are where glycolysis is regulated. We'll understand how the enzyme phosphofructokinase, or PFK, is regulated, and we'll get some structure-function information about that. And we'll understand why ATP increases the cooperativity of PFK. Our Meet a Scientist moment today, folks, is Henry Wedler. He got his PhD in organic chemistry from UC Davis, and he is blind. So his PhD work was some computational work to understand reaction mechanisms. Up until now, we've seen a handful of reaction mechanisms, right? How do we know these reaction mechanisms? Well, thankfully, we have computational chemists like Henry, and we have chemists who actually perform wet lab experiments to confirm the reaction mechanisms, which is pretty cool stuff. So first, let's talk about anaerobic fermentation, which happens in our bodies on the regular. When we are working out, folks, our bodies can either choose to make ATP through regular oxidative metabolism with glycolysis, citric acid cycle, electron transport, etc., or it can choose to do anaerobic metabolism. During aerobic conditions, we use NADH. We oxidize it back to NAD. In electron transport. So this is probably going to happen during some type of light exercise, like jogging or other types of low intensity workouts, walking. But what happens when we don't have access to oxygen, or if we need ATP really fast? Well, oxygen is going to be limited during anaerobic conditions. So think like sprinting, think maybe lifting in a way that becomes anaerobic. In these scenarios, we need ATP very fast. Aerobic metabolism is quite slow. So we won't do aerobic metabolism if we need a lot of ATP. We'll do anaerobic. What happens for anaerobic metabolism, or anaerobic fermentation, is that the pyruvate made from glycolysis, rather than going into the citric acid cycle, is reduced to lactate. This helps us regenerate NAD. Recall that in glycolysis, we convert NAD to NADH. Well, if we need glycolysis to continue, we need more of its substrate, NAD, right? Glycolysis can only continue if more NAD is present. So when we reduce pyruvate to lactate, we can regenerate the NAD required for glycolysis. Together, folks, we'll be focusing on what's happening in humans. We will not really be talking about what happens in other organisms. So let's talk about the enzyme that converts pyruvate to lactate. That enzyme is called lactate dehydrogenase, LDH is its abbreviation. It takes pyruvate and NADH and converts them into lactate and NAD+. If we're talking about muscle that needs ATP fast, has a large requirement for ATP, we have to kind of think about what's going to happen. Glycolysis is super fast, but the citric acid cycle and oxidative phosphorylation are slow. If we need ATP fast, we'll probably do anaerobic fermentation because it's super fast. The overall reaction for anaerobic fermentation is conversion of glucose into ADP, into 2-lactate, and to ATP. So you can see this process gives us the ATP we need, only 2, but it'll happen really fast. Let's take a look at this enzyme real quick before discussing regulation, okay? I like to take a look at this enzyme because it's just kind of showing us some recurring themes from mechanisms we've already seen. And here we actually have drawn out the NADH mechanism, which I think is really nice. So let's walk ourselves through it and then look at some different R groups. So the mechanism here, folks, is going to be pretty simple. We have NADH here. Recall that NADH donates a unit of hydride. Hydride is a fantastic nucleophile. So this nucleophile performs a nucleophilic attack on the carbonyl carbon. Now we also have addition of a proton in the mechanism, right? So probably what's going to happen is we have something, like this take place, where we use histidine, for example, fantastic acid base catalyst, to donate a proton to convert pyruvate here to lactate, where you see that oxygen has been protonated. What else do we see? Well, histidine, if I erase those arrows real quick so we can just take a look, that histidine there looks like it's performing some nice hydrogen bonding interactions with the carbonyl oxygen. But you'll see also if we have a nucleophilic attack here, we'll have a negatively charged tetrahedral intermediate. That histidine is likely also performing charge stabilization of the reactive intermediate. We have additional charge stabilization through this positively charged arginine, and even additional charge stabilization through this other arginine in the active site. Of course, pyruvate is a negatively charged molecule itself before we even start the mechanism. And that is our introduction to anaerobic fermentation. Let's talk a little bit more about regulation of glycolysis.